I'm pleased to introduce to you our speaker, Dr. Hamilton. Dr. Hamilton has been investigating neuroscience associated with chemoreception nearly 35 years. Currently, she's an associate professor in the Department of Cellular Biology and Anatomy at Louisiana State University Health Sciences Center, Shreveport. She is currently funded by NIH to investigate the structure, function, and plasticity of the olfactory system with the goal of understanding the mechanisms that permit us to detect and discriminate such a wide array of odors. So please join me in giving a warm welcome to Dr. Hamilton. Thank you.
Thank you, Corey. Okay, if I can just figure out how to use this mouse, we'll be okay. So I'm going to do a couple of things today. I'm going to try to convince you that the olfactory system is worth studying. It's recently been predicted that we can detect and discriminate on the order of 1 trillion odors. That's an awful lot. And as you may imagine from this picture, it takes a good nose to do that. You have to have a very functional nasal epithelium where the sensory cells reside. What you might not realize, it also takes a great deal of neuronal processing by relatively simple networks in the brain, beginning with an area that I study called the olfactory bulb. Now, I didn't start out studying this. I started out as a marine biologist. So I would like to just take you through that history a little bit, as well as talking about my current research today and how I got where I am now. Okay, so this is the outline. I'm going to start by talking a little bit about how neurons in the brain encode odor information. At least this is how we think it works. And then I'll move on to these other topics. So the olfactory system has been targeted for research by neurophysiologists, people like me that record from neurons, since the very earliest days of recording. This is from a quote by Lord Adrian, who was an early neurophysiologist who began his work in the 40s, and he was quite famous. And he recorded from cells in the olfactory bulb called mitral cells. They look like a bishop's mitre. They have long dendrites that go crosswise here and aren't shown. There are smaller versions of these called tufted cells, and they're the only output from this olfactory bulb to other parts of the brain. Now, in the olfactory bulb, there are also structures called glomeruli, and this is where the axons of the sensory neurons in your nose project through the skull into these glomeruli, and they synapse there with the mitral and tufted cells. Okay, so this is input, this is output. So sensory systems are attractive for studying because you know what the input is, it's an odor. And you can measure the output by recording action potentials, and that's what this is showing here. These are responses of the same mitral cell to different odors. Okay, now the complication is that there are large numbers of cells that I've shown as little star-like things called interneurons. These don't project out of the bulb, and they're inhibitory. They inhibit the output cells, and they do that in the glomeruli and in a layer that I'm going to discuss in a minute. And there are very large numbers of these cells. And anyway, so after studying this relatively simple neural network, Lord Adrian said, and this was near the end of his career, although everyone knows that many animals rely on the sense of smell far more than we do, I am bound to say that until I began to work on the olfactory organ, I had no idea what a large and complex affair it might be. Many, many years later, I would say this is still quite true. We know a lot more about how it works, but we still don't understand how people can detect and discriminate so many odors. But I'll tell you how we think it works. Okay, so we use a simpler model for most of the work. It's a mouse. And we know that mice have about 1,000 genes that generate olfactory receptor proteins. These are the proteins expressed by the sensory neurons that actually detect odors. Okay, and then they generate action potentials as a result. And these genes are each olfactory receptor cell has one gene only. Oh, thank you so much, Barbara. I appreciate it. I have my usual morning allergy cough, which is worse in the library, I think. Anyway, there are 4 zones where these, in the olfactory epithelium, where these genes are expressed by neurons. And the interesting thing is that the neurons that have the same gene project their axons and somehow come together and innervate 1 or 2 glomeruli out of a population of about 2,000. So if you look at the ratio, that means that there's 1 gene in 2 glomeruli. Okay. We're pretty sure that mice can discriminate far more than 2,000 odors. So in humans, it's an even more obvious what's going on. And we think that what's going on is that what the odors discriminate is different side groups and chain length of different types of odors. They're basically chromatographs, like chemical detectors looking for the signature of a particular configuration of a molecule. So we think it works like this. If olfactory receptors, that I'm calling yellow here, detect odors that look like this, they would respond best to something that's rancid, sort of fatty, burnt smell. Now, if they look a little bit like this, no, the oxygen is missing here, but the chain length is similar, it's a little bit longer. They would respond to green and red odors, which together smell like sweet, herbal, woody. This is an alcohol. They tend to be floral in their odors. And then if you put rancid and alcohol together, like this, a longer chain, you would detect violet as the strongest note. So we think this olfactory bulb is sort of like and the glomeruli are sort of like the LED big giant board in Times Square. So different patterns of these glomeruli will light up when signatures of odors are detected. Which ones and where they occur are important because the space of neurons, how they're distributed, is used to generate this sort of board that lights up. Okay. So there's clearly not a one-to-one relationship between a gene and an odor. I thought I had a thing about humans here. I must have, did I skip it? Yeah, I skipped it, I guess. Anyway, took it out. We actually only have 350 functional olfactory receptor genes, far more neurons than the mouse. Now, how do you get 1 trillion odors out of that? So it has to be a very highly combinatorial system. Okay, so let's take a look at what Lord Adrian said was a relatively simple system. Okay, so these are 2 glomeruli that are close together. The big cells here are the mitral cells. They have very long dendrites that run perpendicularly to the glomeruli. And there are smaller versions of those cells called tufted cells. And they have fairly long dendrites that run perpendicularly to the glomeruli. These different cell types define strata or laminae between the glomeruli and the cell bodies of the mitral cells. And this region is called the external plexiform layer, or EPL for short. And it has substrata that are defined by these dendrites. Now, in there, we know they interact with dendrites cells called granule cells, which also have fairly long dendrites. So the interesting thing is, in recent years, we've been able to record from these really small cells that are interneurons. They were originally identified by early anatomists and called Van Gehoekten cells. I just say VG. Or now we tend to call them multipolar cells because they have dendrites that just go in all directions. Okay, no output. Okay, so I became interested in them because we think that the spatial relationship of the glomeruli that are excited by odors and the mitral cells and tufted cells that project that excitation to other parts of the brain has to be limited in space to clusters of glomeruli that detect similar odors. And the only cells in the bulb that can do that, maintain sort of columnar excitation between here and here, are these little cells. And all over the brain, they have been hot area of investigation in the last 15, 20 years. Previously, we really kind of ignored them because they're small and they're hard to work with, and they tend to not be very, they're rather diffuse in their distribution, so you don't see lots of them at one time. So they're hard to study. But we think that there are zones that are related to the glomeruli that are maintained in the olfactory bulb external plexiform layer by these little interneurons, and that's what I study. And we'd like to know how they might do that. Okay, so now everyone should think about why what they're doing is important in science. I think it's important just because it's fascinating. I'll be quite honest. I mean, even if it didn't have any predicted benefit towards human health or, designing new rocket ships or whatever, whatever, new space-related gadgets. It's still important just because it's there. It's like Everest. We want to climb it because it's there. And it's a fascinating part of the brain to me. And it's very important. People don't realize that because you can do okay without a sense of smell. But people who lose it, especially the elderly, are not happy without it. It really affects the quality of life, and it's a serious problem in places like nursing homes. People lose the interest in eating food because they don't have a sense of smell anymore. Most of what your flavor discrimination is smell. It's not taste. You lose smell, what are you left with? You want fatty, crunchy, salty, sweet things that stimulate your taste buds and your GI system. Not very healthy. Anyway, so there are other reasons. We know that olfactory deficits occur very early in quite a few disorders. Parkinson's disease, Alzheimer's disease, frontotemporal dementia, schizophrenia in your teens. And there are kits for trying to identify how good your sense of smell is, and they are used in clinics to examine olfactory disorders. We know that in the external plexiform layer, there are things called Lewy bodies and other things that occur quite early in Alzheimer's disease, and there are other changes in Parkinson's disease. So there is an interest in understanding what kind of synaptic processing goes on. What do the cells do to interact with each other in the external plexiform layer that is interfered with by these diseases? And maybe if we can understand that, we can use this as a predictor in developing new kinds of tests for monitoring these disease processes or in identifying them very early, even earlier than we can now. Okay, so now I'm going to take before returning to mammalian olfaction, I'm going to tell you how I got to this point. And I literally grew up on a dock in the bay on the Gulf Coast of Florida. I mean, you know, in those days, we didn't have iPhones and video games, and, you know, we had to go outside and play. And fortunately, I lived somewhere with a dock and a little boat, and I was fascinated by all the animals that hung out by the dock. Are any of you from down south grow up on the beach or playing in the water? Well, you see that, I mean, it's readily important that to crabs in particular, which tend to hang around docks, the sense of smell is very important, you know. People used to just fish for crabs with a piece of chicken or whatever, and they'll grab onto it, and they're very tenacious, and they'll come right out of the water with it, but they have an excellent sense of smell. So I began my earliest studies as an undergraduate, much like you, and I think undergraduate research is very important because it helps to guide people in choosing their careers and determining whether they like to be scientists or not. So I did this at Florida State University, and at that point I studied parasites of sharks and rays that come from eating snails. It was basically another excuse to go to the beach every couple of months and collect things. But it was a nice study. I got a publication out of it. But I worked on it for 2 years, not just one summer. So, and so that encouraged me to go off and do a doctoral dissertation, and that was at the University of California, Santa Barbara, another place with fabulous ocean views and, you know, a place where you can readily surf and play in the water. And I studied crab chemoreception, and I looked at how these structures called setae, they're hairs. Crabs are actually quite hairy, and they contain dendrites. This is an electron micrograph showing the dendrites of the sensory neurons underneath one of these things. And in this particular crab, the surface structures get worn away. And I wanted to know if they could still discriminate anything in terms of chemicals. And it turns out as long as these dendrites are still present, they respond to all kinds of things. They respond to mechanical stimulation as well as lots of chemicals that are found in seawater. So they were quite functional. So even then, I was interested in chemical senses from the very beginning. Now, I should tell you that I wasted 3 years in graduate school. It wasn't a waste. I studied a deep sea crab that had to be caught by going on long trawls. And it disappeared because of an El Nino cycle. And I wanted to graduate, so I switched to something on land that I could go collect myself. These crabs live in the intertidal zone. The other reason I did that is because I am horribly seasick. Nothing's worse than being at sea for a week at a time in big swells on a ship that's just going up and down like this, parallel to the waves, dragging a big net through the water column. So anyway, I'd already decided I was not cut out for oceanography. But I like being around marine creatures. And so from there, I did a postdoctoral fellowship at another nice marine place. This is a lab in Florida that has quite a few researchers studying chemical senses. And I worked on spiny lobsters, which are, and at this point, I moved into the central nervous system. I wanted to know how these spiny lobsters integrate chemical senses and other mechanical sensory inputs in the brain. So this is a drawing of a lobster brain, and these are recordings from neurons in the brain. And the benefit here is that we only use the heads. So you can imagine what we did with all of the tails. It was a tasty postdoctoral experience. Okay, from there, I decided I wanted to move to the big city. And so I went to Boston. And it wasn't easy to collect anything to use for research in Boston. So we used an amphibian called a tiger salamander that are pretty common in Louisiana. These are actually the animals I enjoyed working on best. So this is a picture of the Chinatown area of Boston. Tufts Medical School is right here. I worked at the New England Medical Center, which is the med school portion, and I actually, quite by accident, lived in this house and only had to walk about 4 blocks. But anyway, we studied both the sensory neurons in the nose, this is a picture of one, and mitral cells in the olfactory bulb. Again, using different odors and concentrations of odors, and noticing that you get these different patterns of action potentials that depend on the concentration and the type of odor. Okay. So here I really became interested in the brain. What's it doing to process all this information, to generate these different patterns, and what do these patterns mean? So I continue my career here, and most recently, I've switched to mice. Now, there are a couple of reasons for this. The first is that someone had already shown in mice, these are the glomeruli, and the big mitral cells are down here, and in between there are all these little tufted cells and interneurons. And they used a virus to trace connections from cell to cell. And they showed that there were these columns of neurons that were labeled together. Although there's no obvious columnar arrangement, there is a functional columnar arrangement that appears to be related to the glomeruli. So I've been studying this using a mouse that, in which the interneurons express this fluorescent green protein, enhanced green fluorescent protein. If the neuron has an enzyme called GAD65, it's glutamic acid decarboxylase 65 kilodalton isoform. There are 2 isoforms. And basically, GAD generates GABA. So neurons that are inhibitory interneurons have to have one of the 2 GADs. Okay? So if you can make most of the GADs, this particular type is found very much in the external plexiform layer of the olfactory bulb. If you can make them green, you can see them. So then you can make a brain slice and look for green cells and say, okay, there's one, so you can record from it. And what we've done here is stain all the nuclei with something called cytox. So the yellow cells have both. So these are the different layers of the olfactory bulb, and this area in between here is the external plexiform layer. So we've been recording from those cells, and we've been examining the functional properties of the neurons. So I'm not going to go into any details here. I've got way too many slides. But basically, we record from one of these little interneurons, and then we record from mitral and tufted cells that are nearby. And we ask the question, is the activity of one of the little interneurons correlated in time with the activity of any of these other mitral and tufted cells? And so you just record. That's all you do. Now, I have a very talented colleague who can record from up to 4 at one time, which is no minor feat. And so the interneurons have these spontaneous potentials called excitatory postsynaptic potentials. And this indicates that they're being excited by other neurons that are presynaptic, the feeder neurons. And so we want to know, are they tufted cells? Are they mitral cells? So you record from all the neighboring cells that are kind of in the same column. And this picture shows where the cells were. The interneuron, I think, is this one. Yeah. And then 4 is a mitral cell. 2 is a mitral cell. No, this is a mitral cell. I think 2 is, where's this? This is a tufted cell over here. I'm having trouble seeing this from this far away. I'm very nearsighted. Anyway, so you do these recordings and you use a statistical measure of the probability that action potentials in the mitral and tufted cells are correlated in time with the postsynaptic potentials. And you get graphs that look like this. And the bottom line is, we know that, yes, they are coincident, at least in terms of big bursts of activity, with activity of mitral and tufted cells that are nearby, sort of in the same column. And we know that that not only that, they are monosynaptic, and that's getting a little complicated, but basically what it shows is that the excitation of the mitral cell can directly excite one of these little cells, sort of a one-to-one relationship. So we think we know where the input's coming from. We know something about its unique characteristics in terms of the receptors involved, and this sets these neurons apart from other neurons in the olfactory bulb. So the next thing to do, and I'm going to have to hurry up, I guess, because I'm going too long, you want to know what the cells look like. You know, how are they related spatially to the glomeruli? So you inject a dye when you're recording. So this is an interneuron, and we had a dye in the pipette. And this is what the dye looks like. It's red. And if you can tell using a confocal microscope, which can make thin optical slices so you can image the cell in 3 dimensions, and this is just the analysis of that, we can show that this cell does in fact have the GFP. It's not 2 cells sitting on top of each other, it's 1 cell. Okay? So when we see that, then we know, okay, let's draw it. This is actually the hardest part. It can take a week. And you do it with a computer, and it's very painstaking. And you end up with a branching pattern like this, and then you analyze that, and you get a drawing like this, and all of these different branches are measurements, first order, second order, third order branch, how far from the cell body, how far from the different layers of the olfactory bulb, and you end up with a lot of data. And so then you have to do something called principal component analysis, and from there you do something called cluster analysis. And after many reiterations using those programs, you end up with a statistical measure telling you whether cells really look like each other or not. Because this is a 2-dimensional drawing. The bulb is a round structure, so you have to analyze all of this in 3 dimensions. And so basically we found that there were 4 different categories of these cells based on their branching patterns. So we think there are at least 4 subtypes, and they clearly define different amounts of glomerular space in the EPL. So we predict that some subtypes will work mostly with one type of tufted cell and the others with maybe both mitral and tufted cells. So that's where we are today, is trying to sort that out. Okay, so this is just a summary of what I've told you. Now, I'm going to take just a few minutes and tell you about something that's totally different. And this is looking at the nasal epithelium, and in fact, the olfactory portion of the epithelium of these mice. And we did this quite as a control. And one of my students kept saying, there are green cells in the nose. And I kept saying, no, there aren't, because there are no GABAergic, no interneurons, no inhibition in the nose. They're all just detectors, and they send all that information to the brain. Now, there are glands out there that are innervated by your autonomic nervous system, but they don't use GABA as a transmitter. So there should be no GAD in the nose. There's no reason for it to be there. So I thought he was doing something wrong, and lo and behold, I finally looked, and by golly, there are green cells in the nose. And they sure look like sensory neurons. They have a little knob, a long dendrite, and a cell body. And we've been examining those more, and it turns out they're immature. They don't ever become mature sensory neurons. And we knew that by staining for a protein called olfactory marker protein that's only found in the mature cells. We know these cells are also immunoreactive for proteins that are only found in the immature cells. And we know that their axons, this is a bundle of axons deep in the olfactory epithelium in the nose. You can see there are green ones coming from these cells and red ones coming from the mature cells. And they all get bundled together and they go through the skull into the olfactory bulb. So we know they get there, but they're not mature. So what are they doing? Do they have GAD? And if so, is it some point in the life cycle of these cells, are they generating GABA? Why don't they ever mature? Well, we know that they stay in the cell cycle by staining for something called Ki67. You can tell that these cells are, in fact, still active in the cell cycle. They don't seem to die because we don't see many that have this enzyme, which is involved in self-death, apoptosis, or apoptosis, some people say. So if you want to know more about this, you'll have to go to the LBRN poster session and see Christina Oak's poster. We've been trying to determine if there is any GAD in the olfactory epithelium, and learning how to do RT-PCR in one summer is a big challenge. So we know what we're doing, but we still have some problems with getting the tissue right. So anyway, Christina and Olivia here have worked very hard on this. So these are the people I need to thank today, and I want to especially thank Christina and Olivia Washington. Olivia is in a program that we have for high school students at the Med Center, and they've been working very hard, and I appreciate their help. So thank you very much, and I suppose I have time for a few questions or not?
Yeah, we do.
Okay. Anybody? I know that's a lot to go through in a hurry.
Let's go ahead and start. Any questions from your location?
Come on. There are no stupid questions. Yeah.
So since mice have some sort of neurons in their olfactory system, did you look at humans and did you see the same results?
No one has been able to record from the human.
Can you repeat the question for us?
I'm sorry. The question is,
okay.
have we looked at how olfactory information is processed in humans? Not in live humans. You can't really do that. People have recorded from the olfactory epithelium, and you see the same kinds of recordings you get in any vertebrate, all the way from a fish to us. I should have mentioned, this is a relatively primitive part of the brain. In fact, it's a very primitive part of the brain. It's found in all vertebrates. It looks very similar in all vertebrates. Almost all of them have mitral cells, ducted cells, and interneurons of various sorts. So we think that a mouse is a good model. There are differences between mouse and human, obviously. Rodents use their olfactory sense for almost everything. We can get along without it. We're more visual. But as I said, it's not, people aren't happy without it. It's a quality of life issue. Yes?
You said that there's a loss of, cells with Parkinson's disease and Alzheimer's?
Yeah.
Do they know what the reason behind that is?
The question is, do we understand why the sense of smell deteriorates so quickly, so early on, in Alzheimer's disease and Parkinson's disease? All I can tell you for sure is that we know that in Parkinson's disease there is a loss of neurons that results in a motor deficit and eventually, and also cognitive deficits that people don't often think about. We know that the the neurons that are lost in Parkinson's disease are dopaminergic. They have dopamine as a neurotransmitter. We know that in the olfactory bulb, there is a population of those neurons and they are not lost. But there are definite changes in terms of what dopamine is doing there. So the synaptic network is being affected. We don't understand exactly how. And the same is true for Alzheimer's disease. We can see the pathology of Alzheimer's disease very early in the human olfactory bulb, in the epithelium. But exactly why that disrupts and how that disrupts the circuitry is not known. Yes?
I know you mentioned that climbing the mountain didn't necessarily directly show the human, but could this olfactory model be used to shed some light on some things that might be happening in Alzheimer's and the actual biochemistry of it? I know a lot of this was sort of electro.
Yes. Yeah. The question is, can the model that we have working with mice be used to shed some light or help us understand what happens physiologically in Alzheimer's disease and Parkinson's disease? Is that what you were asking? Well, we think it will, because we know the neurons that are affected. At least we know the location of the neurons that are affected. We don't know how they're affected. Now, people have tried taking nasal biopsies in Parkinson's disease, and that holds some promise. But it's if you've ever had a nosebleed, you know that routinely you don't want to go around taking little bits of epithelium out of the nose. But it's possible that there may be other tests eventually linked to somehow nasal swabs or something that's less detrimental. And it could be that, for example, classes of odors are lost first. So we just need a whole lot of study on those issues. It's compounded by the fact that, like everything else, you lose acuity with aging. There's a tendency for dementia with aging. So it's compounded by those problems. It's a tough thing to do, but there's a great deal of interest in trying to do it. Anyone else?
We'll
Okay.
ask New Orleans if they have any other questions. Does the New Orleans site have questions? Just speak up. Okay. Well, then we have a couple questions over here. Sabine, would you like to?
I was just wondering if you were familiar with this study about the olfactory cells within the digestive system and how they help detect poisons when ingested, and if that epithelium is at all similar, if it's connected to the same part of the brain.
I haven't heard about the poison studies. I mean, most poisons are bitter, and you have very strong sensitivity to bitter in your mouth. So bitter basically tells you to spit it out, not swallow it, unless you learn through training that some bitter things are good. Like, babies won't eat mustard, but they learn to like it eventually. But anyway, what I do know about the studies in the GI system is that they detect fat. So low-fat diets don't work because your GI system is somehow telling your brain that it's not getting something you need. Unfortunately, it's way too prevalent in our diet, and we don't have a way to damp that down because the theory is in evolution there was not enough fat, so you need fat. And so I don't know about bitter, but I do know there are taste cells in the GI system. And exactly how they're wired, I think is still debatable. But I'd be happy for you to fill me in on this.
I'm not exactly sure what the name of the study is. I just remember reading it in one of my physiology classes, and it was just olfactory cells within the epithelium in the GI tract that detected
they're there.
bitter.
And we didn't know about them 20 years ago. So.
Yeah, they cause the cilia within the digestive tract to beat faster and to move the bitter substance through faster.
Okay.
I don't know. I was just wondering.
Was that in Dr. Caprio's class?
No, I don't go to LSU.
Oh, okay, sorry. There's a very famous fish physiologist in Baton Rouge who studies catfish smell and taste.
Okay. Do we have any other questions? Anyone? All right. Well, let's everyone thank Dr. Hamilton for her talk.
Thank you.